So after the Jonah and cortical reactions, the cytoplasm of the sperm enters into secondary oocyte. After the entry of cytoplasm of sperm into secondary oocyte through zona pellucida and plasma membrane, it induces completion of meiotic division of secondary oocyte. Here you can remember one point. At the time of we discussed the process of oogenesis. The oogenesis final step. So what we discussed, secondary oocyte undergoes meiotic division. Which meiosis? Second meiotic division, and arrested at metaphase 2 and completed at the time of after the sperm entry at the time of reproductive life. So once a sperm enters the cytoplasm of the sperm enters into the secondary oocyte, the completion of meiotic division takes place in the secondary oocyte, and what is formed? One second polar body, and finally ooted formation takes place. That is actual ova. See here, the secondary oocyte undergoes meiosis 2, and after the sperm entry at the time of reproductive life, formation of one ooted and one second polar body. The ooted fusion with sperm, and finally the formation of zygote, right? Okay, so here there is ooted formation takes place after the cytoplasm of sperm enters into the secondary oocyte, and the ooted is ready to fusion with the cytoplasm of sperm. So mixing of chromosomes of a sperm and an ovum is called karyogamy. Karyogamy. So what is the next step? After sperm entry, karyogamy takes place. Karyogamy. What is karyogamy? The fusion of chromosome material of sperms and ova is called karyogamy, or also called as amphimixis. Karyogamy are also called as amphimixis. So after the fusion of chromosome material of sperm and ova, what is formed? Zygote formation takes place. Diploid zygote formation takes place. So after karyogamy, so what is the next step? Activation of egg. So here, finally by the fertilizing and anti-fertilizing interaction, zygote is formed. So here there is fertilizing and anti-fertilizing interaction takes place. Same species of egg and sperm only forms zygote. For example, in human female, the sperm of a dog enters in case it is possible for fertilization. The fertilization is possible? No. What is the reason? The reason is finally at the time of formation of the zygote, the fertilizing and anti-fertilizing interaction takes place. Same species of egg and sperm only can be fertile, and finally zygote formation takes place. Same species by the fertilizing and anti-fertilizing, and zygote is formed by haploid sperm and haploid ooted zygote. Diploid zygote 2n is formed by haploid sperm N and haploid ooted N. Alright? Okay. So what is the chromosome number? 2n, 46. What is haploid? 33 and 33. Right. So this is regarding about fertilization and different types of events of fertilization. So once again, we can revise the topic of fertilization. So what is meant by fertilization? The fusion of sperm and ova to form zygote process is called fertilization. Otherwise, the diploid zygote, the process of formation of diploid zygote from the fusion of sperm, from the haploid sperm and haploid ova, fusion of haploid sperm and haploid ova is called fertilization. So when fertilization occurs, before insemination or after insemination? After insemination. So where the sperms are released in the region of vagina of the females, and sperm passes through the cervix, uterus, and finally reaches the ampulla and isthmic junction of the fallopian tube, right? So in case simultaneously ovum is released from the ovary, at that time the sperm centrifuge takes place, then only the fertilization is occurs at the junction of ampulla and isthmic junction, at the region of ampulla and isthmic junction. So where the fertilization is occurs, ampulla and isthmic junction. So that only all copulations or not leads to fertilization and pregnancy, right? And how much sperms are releases? Approximately there is 300 billion sperms are releases at the time of copulation, but all sperms are not going to be fertile with the secondary oocytes. What is the reason? Because of a majority of the sperms are engulfed by leukocytes of the vaginal epithelium, right? And how much time is required for the fertilization after the releasing of secondary oocyte? That is 24 hours. After 24 hours, the ova is not fertile; it is going to be degenerated. And the sperms in the female genital tract are the capability of the fertilizing the egg by the secretions of the female genital tract is called capacitation. So when capacitation is occurs by the sperms after insemination in the region of female genital tract by the secretions of the egg. So physical and chemical events of fertilization: acrosomal reaction, cortical reaction, sperm entry, karyogamy, and activation of the egg. By acrosomal and cortical reactions, sperms the secondary oocyte walls envelopes are going to be dissolved, and sperm cytoplasm of the sperm enters into the secondary oocyte, and finally karyogamy takes place. So what is karyogamy? So karyogamy or amphibixis. So mixing of chromosome material, sperm and ova is called karyogamy, and finally zygote is formed, and the secondary oocyte accepts only monospermy, not polyspermy, because polyspermy means entry of more than one sperms into the ovary. It is not possible, right? One sperm into the more than one sperm into the ovum, not ovary, right? Okay. So next, so finally, fertilization and anti-fertilization interaction. By this interaction, zygote is formed. Same species of the egg and sperm only formed. So secondary oocyte undergoes meiosis II, and one ooted and one secondary polar body is formed, and ooted with fusion with the sperm, and finally zygote formation takes place. So this is regarding about fertilization. After that, we can go through the NCERT lines and the diagram of ova given in NCERT. Thank you.